A common goal is a healthy, long life. To obtain that, we typically read on the subject and watch videos, among other things. Something we run into quite quickly is contradictory information, which leads us to look for corroborating and refuting evidence. Let's cover a broad outline of useful health and longevity research parameters to help with that and other aspects of interpreting scientific claims. Welcome to ForGevity, where we focus on topics in healthspan and lifespan applicable to humans. Coverage spans practical nutrition, supplementation, physical and mental fitness, sleep, and other topics, along with empirical strengths and weaknesses of various protocols for all types of people, sex, age, condition, etc. The topic of this video series is health and longevity research parameters. We often hear influencers saying to do or don't do, eat or don't eat, or take or don't take this or that. Few reasons may be given. Imagine you scroll through your feed and see a post claiming X cures Alzheimer's. Before you decide to share, ask yourself, what did the actual studies say, and how solid is the evidence behind that headline? How do we judge the advice? To start with, we suggest never automatically trust the headline. The headline might say, X does Y. However, the first paragraph of the report often says, X may do Y, which means X may not do Y, a far cry from does. Furthermore, that first paragraph might represent a misinterpretation of the study altogether. Even well-conducted studies can mislead if interpreted without sufficient context, something we'll explore later in this series. The full study might not even suggest that X may do Y. What's the quality of any research backing up the advice? We can take steps to help reduce the likelihood of building upon false premises. Read, analyze, and critique the full study in the light of what's covered in this video series. Definition: critical appraisal. A systematic evaluation of a study's design, methodology, and reporting to identify strengths and weaknesses. Why it matters: detecting flaws in randomization, blinding, or statistical analysis prevents reliance on invalid or biased results. We'll explain these things in greater detail in this series. Research any claims in terms of all-cause and cause-specific mortality. Does the suggested action or inaction reduce mortality in general? All-cause mortality refers to death from any cause, not just the disease or condition being studied. Cause-specific mortality refers to deaths attributed to a particular disease or condition. Why it matters: a treatment that lowers cause-specific mortality might not improve and could even worsen overall survival. For example, a diabetes drug might reduce cardiovascular deaths but increase cancer risk, leaving overall mortality unchanged or worse. Looking at all-cause mortality provides a broader and more honest picture of health impact. Does it extend health span? That's the years spent in good health, free from chronic disease and disability. Extending lifespan without improving health span could mean more years of poor quality of life. The concept of health span does not necessarily include aging itself. Traditionally, aging has not been classified as a disease. However, the emerging field of geroscience increasingly treats aging as a modifiable risk factor or even a disease in its own right. Understanding whether aging is considered a disease has implications for how studies define and measure health span, whether they're tracking biomarkers of cellular aging, absence of chronic conditions, or functional capacity like mobility and cognition. If aging is seen as a disease, interventions might shift toward targeting fundamental aging processes rather than just disease-specific outcomes. Now that we've briefly covered critically evaluating headlines, study claims, mortality, health span, and research quality, let's break down the hierarchy of evidence from weakest to strongest and then cover other key research factors. Here's a hierarchy of evidence from weakest to strongest. Anecdotal evidence. Definition: informal stories or personal experiences shared by individuals, often without systematic data collection or analysis. Example: a friend's account of improved energy after taking supplement X, or a blog post describing a dramatic weight loss journey. Why it matters. Anecdotes can spark curiosity and highlight potential effects, but they lack controls, are susceptible to bias, and cannot establish cause and effect. They serve as a starting point for hypothesis generation rather than proof. It could be coincidence. A random fluctuation or unrelated factor explains the perceived effect. Placebo effect. Belief in a treatment's efficacy leads to real or perceived improvements. It may have actually done something beneficial, but how long will it last? Short-term observations may not translate to sustained benefits. Expert opinions. Definition. Recommendations or conclusions drawn by individuals with recognized expertise or credentials in a field, often based on experience, clinical practice, or interpretation of existing data. Why it matters. Expert opinions can guide preliminary understanding when empirical data are scarce, but they may reflect personal bias or conflicting viewpoints. Critical appraisal of the expert's background, potential conflicts of interest, and the evidence base supporting their opinions is essential. Computational studies. Definition. Research using mathematical models, algorithms, and large data sets to simulate biological processes, predict outcomes, or identify patterns without direct experimental manipulation. Why it matters. These studies can generate hypotheses and interpret complex data at scale, but their conclusions depend on model assumptions and the quality of input data. Validation through experimental or clinical studies is necessary. Interventional studies. Definition. Research where investigators actively assign participants or samples to different treatments or exposures to test effects under controlled conditions. Why it matters. They can establish causality and measure specific outcomes, but design quality, randomization, blinding, control groups, and ethical considerations affect reliability. Subtypes include in vitro studies, quote, in test tubes or culture dishes, close quote, experiments using cells or tissues outside a living organism. Why it matters. Allow precise control of variables and mechanistic insights, but lack whole organism complexity and systemic interactions. In vivo studies, quote, in the body, close quote, experiments conducted in living organisms, animals or humans. Why it matters. Captures systemic effects in pharmacokinetics, absorption, distribution, metabolism, and excretion of drugs, but can be limited by ethical constraints, species differences, and higher cost. Cell studies. Definition. Experiments on isolated cell lines to investigate molecular or cellular responses to interventions. Why it matters. Offer mechanistic detail at the cellular level, but may not reflect tissue or organism-level processes. Summary of Part 1. In Part 1, we laid the foundation for navigating the complexities of health and longevity research. We examined the pitfalls of relying on headlines. We outlined how to critically assess study quality, asking whether an intervention improves all-cause mortality or merely changes one cause while worsening another. We also discussed the importance of Healthspan and how evolving views of aging as a disease may shift the way studies are framed. Finally, we began breaking down the hierarchy of evidence, from anecdotes to interventional studies, setting the stage for deeper dives in the rest of the series. Please pause the video to like, share on social media, subscribe, turn on notifications. See the video's bibliography and links in the video's description. Leave a thoughtful comment and engage with others in the comments section. Then, unpause the video for any further information. Thank you. For this channel, one size does not fit all. In addition, civility between dietary camps or tribes, vegans, carnivores, and others, is encouraged. ForGevity deliberately avoids a know-it-all approach. Settled science often becomes unsettled upon new research. ForGevity may further the process by raising new questions for researchers and scientists to address. That concludes Part 1. Join us for Part 2, where we'll continue research parameters and pick up by explaining model organisms. Disclaimer: no medicine is practiced by ForGevity. Materials provided are for educational purposes only and are not intended for medical advice, diagnosis, or treatment. Please consult a licensed medical practitioner. Credits: research, script, and audio/video development by Tom Usher. Copyright 2025 by ForGevity. All rights reserved.